Hey everyone, and welcome back to the Biochemistry YouTube channel. My name's Jess, and today we're going to talk about the Wobble base hypothesis. So the Wobble base hypothesis is going to occur during translation when the first two bases of the mRNA codon demonstrate Watson-Crick base pairing, but the third base varies from this base pairing. When we talk about the codon and anticodon, we are assuming that codon base pair number 1 is on the 5' end, and base pair number 3 is on the 3' end of the codon. When we're referencing our tRNA molecule, we assume that base pair number 1 is on the 5' end, and base pair number 3 is on the 3' end. So when we talk about this third base varying, this should logistically make sense looking at our genetic code. Look at how many different sequences can code for one amino acid. Remember that our genetic code is degenerate. Notice how these sequences have the same first two codons, but they vary at the third codon. Let's take a look at the different Wobble base pairing that can occur. Let's look at this mRNA and tRNA molecule setup here. So our Wobble base is going to be in this position here, our third position of our codon and our first position of our anticodon. Say that guanine is in the third position. It will base pair to cytosine, but it will also base pair to uracil in the Wobble base position. Let's say we have uracil in the third codon position. It can base pair with adenine, but it can also base pair with guanine and inosine. With adenine in the third codon position, it can base pair to uracil and it can base pair to inosine as well. Finally, let's say that cytosine is in our third codon position. Cytosine can base pair with guanine and also inosine. Here is the official chart that was presented in lecture of everything we just went over. Make sure to pause this video and commit to memory the different Wobble base pairings. Let's do a practice problem to wrap it up. Suppose a template strand of DNA reads 3' C, G, A, and 5'. With regards to the Wobble base hypothesis, what tRNA will pair with the corresponding mRNA codon? So how do we solve this problem? We're going to need to understand what our template strand of DNA is. Remember that this is the template for our mRNA, so these sequences are going to be complementary to one another. For step 1, we're going to find our mRNA strand. So if our template strand of DNA reads from the 3' to 5' CGA, that means that our mRNA molecule will read from 5' to 3' GCU. With our mRNA codon, we can find our anticodon. So step 2, we're looking at our mRNA molecule, right, of CGU. We know that our anticodon will be from 3' to 5' CGA. This is with our Watson-Crick base pairing. But this question is asking about the Wobble base hypothesis, so we need to see what else can base pair to U. Using our Wobble base knowledge, we know that U can pair with adenine, but it can also pair with guanine and inosine. So we're going to have 3 answers of possible anticodons here from one DNA strand. Try to answer this question if we were given the coding strand of DNA rather than the template strand, and see if you can determine the answer. That's all for this one, and we'll see you in the next video. Thanks for watching.